Hi everyone, thank you for joining the first webinar in our series, Use of New Approach Methodologies for the Risk Assessment of Pesticides. This series is being co-organized by PETA India, the Biological Agri Solutions Association of India, and the Society for Alternatives to Animal Experiments. My name is Ankita Pandey, and I'm an advisor to PETA India, and I'll be coordinating today's webinar with Mr. Vipin Saini from the Biological Agri Solutions Association of India. Today, we will hear from Dr. SK Jain from Central Insecticide Board and Registration Committee, who will be presenting on use of new approach methodologies to meet pesticide testing requirements in India. Before I introduce the first speaker, I would like to point out that the information about the webinar series can be found at the link on the slide. There will be time for questions for the speaker at the end of the presentation. Everybody is on mute, but you can type your questions or comments in the question section within your GoToWebinar toolbar at any time during the presentation. This toolbar should be on the right-hand side of your screen. So speaking today will be Dr. SK Jain. Dr. Jain received his PhD in pharmacology from the University of Veterinary Medicine, Hanover, Germany in 1994. From 1983 to 1997, he continued to serve as an assistant professor, pharmacology and toxicology with Haryana Agricultural University, Qsar, and later on as a professor at Haryana Agricultural University, Lala Lajpat Rai University of Veterinary and Animal Sciences, Qsar, till 2018, during which he guided 2 PhD students and 9 master students in pharmacology and toxicology. He is a recipient of several awards and fellowships, has 24 research papers published in national and international journals. With 34 years of post-qualification experience, Dr. Jain is currently the Senior Consultant Medical Toxicology with the Central Insecticide Board and Registration Committee, Ministry of Agriculture and Farmers' Welfare. It is now my pleasure to turn the floor over to Dr. Jain.
Good morning, everybody. I am Dr. SK Jain from CIB&RC, where I am working as Senior Consultant Medical Toxicology and looking after the regulatory toxicology for pesticide registration in India. Today, I'll be delivering some important points regarding what new approach methodologies can be adopted for meeting the pesticide testing requirements in India, how the animals use can be minimized for getting the data which can be allowed and approved for registration of various pesticides, including biopesticides in India. In today's talk, we will be covering mainly the different non-animal approaches which can be accepted for pesticide toxicity testing and registration in India, like in chemical, in vitro, ex vivo, and in silico methods. Various integrated approaches can be allowed and can be applied for testing as well as assessment of pesticides. Also, weight of evidence approach, RAD approach or data bridging concept, and AOPs, adverse outcome pathways, can be used for assessing the various pesticides for registration. There are a lot of opportunities where we can waive certain tests on animals and can allow various alternatives for using the registration of pesticides in our country. Now, CIB&RC is promoting that animal use to be reduced and alternates may be allowed, provided scientific and full justification is provided by the registrants. There are a number of guidance documents from various scientific organizations worldwide, which are also accepted in India after evaluation and critical examination. Other toxicity data requirements are also acceptable from other countries, and they are accepted now in India after thorough evaluation. New approach methodologies can be used, and they provide a lot of benefits for minimizing the use of animals for toxicity testing. We'll be discussing in detail in the next slides. Recently, CIB&RC has revised the guidelines for toxicity testing and the registration of various pesticides, including biopesticides, in India. And these guidelines are still under revision, and hopefully they will be applicable and implemented soon after further improvement and revisions. We have already accepted waivers. We are trying to accept much more exemptions and waivers of certain animal toxicity testing. For that, we can allow replacement alternatives where we will be accepting non-animal methods which are used for toxicity testing, like in chemico, in vitro, ex vivo, as well as other methods for accepting the data without animal testing data. These are applicable to both chemical pesticides, and now we have included biopesticides also, where replacement alternatives will be allowed and accepted provided the registrant provides full and sound justification, and that will be accepted case-to-case basis on merit if full data is provided in non-animal methods. We have already one guidance document on toxicology from CIB&RC in India, where we allow lot of waivers and exemptions even without some animal experiments, and we accept the other approaches in which we accept principle of 3 R's, that is replacement, reduction and refinement. This is already known, and it is used world over that animals should be minimized for toxicity testing where we can do it, where it can be possible. The principle of 3 R's of animal use is already there. We are taking it into consideration for registration requirements. It is already recognized by OECD and various other regulatory authorities in the world, and similarly we are also going to accept alternative approaches, including the in vitro test as well as non-animal testing approaches. We also accept waivers, data bridging and in silico models, which provides the weight of evidence approach, and that should be acceptable to the registration committee for grant of registration of various pesticides. For example, there is possibility of combining acute dragon and skin irritation study. OECD 436 is now accepted also, beside the 403 for acute inhalation toxicity study. This is already available and given in the document of 2017 of CIB&RC, that is guidance document on toxicology. So we are considering use of all valid alternative approaches, in vitro methods, etc. There are several non-animal approaches available now, which are internationally accepted and valid, and they are used as integrated testing approach to make health protective decisions in animal studies. We also encourage registrants to use these non-animal methods and approaches, and we also advise that you consider only animal testing as a last resort if you can provide the data in alternative methods, provided it is scientifically sound and it is valid. For example, there are several OECD valid non-animal methods in skin and eye testing. For example, you can see in case of eye irritation or corrosion study, there are various guidelines from OECD 492, 494, 496, 467, 460, 491, 437, 438. These are various models which are used for eye irritation study, where we are using RSC, that is reconstructed human cornea-like epithelium. This is done and used world over, and it has been accepted, I think, in Europe as well as in other countries, and we will be trying to accept, provided with full justification and scientific acumen. We also have EIT, that is eye irritancy test 494. Then we have in vitro macromolecular test method, where we can identify chemicals which can cause eye damage and chemicals not require classification. 467, we have various defined approaches for serious eye damage and eye irritation study. In another study, 460, there is FL test, fluorescent leakage test, which can be used. 491, that is short time exposure in vitro method, is also valid and acceptable. Another animal test, that is in vitro test, bovine corneal opacity and permeability, BCOP test, that is also accepted for eye irritation study. And 438, that is in chicken, isolated chicken eye test is used, and it is valid and it is good test, provided the good results, and that helps avoiding the animal test. Similarly, we have skin irritation or corrosion methods, in vitro methods, which have been validated by OECD 431, 435 and 439. All these tests, they are used in vitro, where we use reconstructed human epidermis. Then membrane barrier test method is also used. In another method, we use RHET method, that is reconstructed human epidermis method. Then we also have various in vitro methods for skin sensitization. These are 497, 442, C, D and E. These four methods, they can be applied for skin sensitization as an alternative to animal test, and the details, they are provided in the website of OECD. The link is given below in the slides, you can see. Then we have further use of integrated approach on testing and assessment, that is IATA, for both skin as well as eye testing. If we go to the website of OECD, there is a document, guidance document on IATA for eye damage and eye irritation, and that is series number 263. For other approaches, we have 256, that is number 256 document is there, that is used for skin sensitization. Then we have for skin irritation and corrosion 203 document is available, and AOPs for skin sensitization, which is initiated by covalent binding to proteins, and the document is number 168. These documents, they are available, and the link is given and on the OECD website, that you can see. So in our view, that integrated approach on testing and assessment, IATA, it has to be applied, and it should be applied for the assessment of both eye as well as skin irritation corrosion study, as well as also the skin sensitization study. And this is, if it is provided with full evidence, we accept it, and we will be accepting it, and CIB&RC is going to accept in future. So we are simplifying the all the guidelines for regulatory toxicology registration. Now we are moving to weight of evidence approach. You know, weight of evidence approach, WHO in 2009, they have defined weight of evidence approach as a process where all the evidence is considered relevant for a risk assessment, both in evaluated form and it is weighted. So it provides the full scientific justification when we accept WOE approach. Then this is similar schematic outline of a typical item, and it should be used in every toxicity. And weight of evidence approach is very important. Waiver would be considered only when existing information will provide the robust and scientifically sound WOE approach, along with data bridging from structurally as well as biologically similar pesticides that will be considered on merit case to case basis. If they provide the sufficient read across data bridging concept data with full weight of evidence, it has to be accepted, and it is going to be accepted in India in future. Now still we are accepting in some cases, but now we want to ensure it to other cases also, for other compounds also, as well as even in new molecules we are allowing the weight of evidence approach for waivers. In this case, the data is examined, both epidemiological and clinical data, as well as the data which is provided by the resistant in animal study, as well as other studies in vitro, ex vivo, or in chemical studies. Even the non-testing strategies, for example, read across or data bridging from the structurally biologically related substances, as well as QSAR, quantitative structural relationship, activity relationship. Now we are accepting in silico models also, but should be scientifically sound and robust. If the data is insufficient, what we can do? We can go for using non-animal methods. We can test the molecule in non-animal methods. If it cannot be done, then we should go for animal testing, and it should be the last priority. We should not use in every case all the tests using the animals. If we can do without animal, then we can go for the alternatives. And there is a series on testing and assessment number 311, which is available. That is guiding principles and key elements for establishing a weight of evidence for chemical assessment. That provides all these details regarding weight of evidence approach. So ultimately, the registrants, the applicants in India, we are encouraging to use them weight of evidence approach, considering all the existing information, as well as if any data is to be generated additionally, then they can also go for non-animal testing, which we have already elaborated. Read across or data bridging. What is read across or data bridging? That we have to consider the similar structure compounds to the target compound, and that will help in finding out whether it is similar or not. In that case, we can accept the data bridging for accepting the data without going into the animal history. So we have uh, guidance on grouping of animals. Second edition series is available, and this is number 1942014. It is already available. Then there is a guidance document of OECD QSAR application toolbox is there for developing the chemical categories as per the OECD guidance document regarding grouping of chemicals, the number 102 2009. The website is given in the slide, and you can have the link and you can see the details, and can be helpful for the registrants and applicants using this red approach and data bridging approach. So ultimately, our aim is that registrants should use this red across data bridging principle for chemically or biologically related pesticides. There they should not go for the animal testing for every purpose. Now we come to the QSAR toolbox. QSAR approaches which are OECD valid, as well as uh, the CATMOS tool, where we can predict the acute oral toxicity. Both can be used and they can be allowed, providing if you submit full data. For example, in case of QSAR toolbox, it has been accepted, it is valid, provided the in silico models provides the full robust data, and that can be accepted for waiver for allowing the in silico model in particular pesticide or compound. Similarly, in CATMOS tool, where we can predict acute oral toxicity, it is a free online resource. It is available for acute oral toxicity, and the work group has organized a global project in silico models, both acute oral systemic toxicity that can predict the endpoints, and it is given in detail. The compound can be very toxic, non-toxic, and the 50-point estimates hazard categories under EPA as well as hazard categories under GHS. The website provides the details, and you can use it for registration of the various pesticides. So now we are trying to encourage the registrants and applicants to use as well as implement valid QSAR approaches, that is in silico can be used for number of pesticides. Then next comes the AOP, that is adverse outcome pathway. What is the adverse outcome pathway? It is an analytical construct that describes a sequential chain of causally linked events at different levels of biological organisms, which leads to an adverse health or ecotoxicological effect, and it can be used to support chemical risk assessment based on mechanistic reasoning. And CIB&RC, CIB&RC in India is encouraging registrants that they can refer to the OECD website, and they can use these approaches in PETA framework. The website is already given, and you can go through and you can use it for risk assessment and for getting the registration. Then So there are a lot of opportunities for exemptions and waivers of the animal testing methods, and various parameters they are allowed without animal testing, and other data can be accepted. So CIB and RC accept waiver requests based on the various following weight of evidence approaches. For example, physicochemical biological properties. That is acceptable in case of acute oral study or eye as well as skin irritation study is not required based on physicochemical or biological properties if the compound is strong acid or it is a base where the pH is less than 2 or it is more than 11.5, and it clearly indicates that it is a corrosive or severely irritant compound, then there is no need for submission of acute oral study or eye skin irritation study, and that can be waived off, and that is allowed on the basis of the property of the compounds. Similarly, in acute inhalation study, that is not required if the vapor pressure is low, that is if it is less than 10 to power minus 2 pascals and it is well known, then there is no need and it is allowed for exemption. There is no need of submission of the data because it's not easy and it's not required. Similarly, if there is technical feasibility is given by the applicant and justified, then for example, acute oral study is not required if the compound is a gas or a vapor at ambient temperature, then there is no need, it cannot be done, and we waive off the oral study. Then acute inhalation study is not required and it is waived off. When the substance is not a gas or liquefied gas, then it cannot be performed in our view, and scientifically it is okay and it is waived off. Then use and exposure patterns. It also helps whether it is required or not. For example, acute dermal study or skin irritation study is not required. For example, if in skin contact is not likely in production or use of the product design prevents dermal exposure. When there is no dermal exposure, when there is no skin contact, there is no chance of any contact with the skin of the compound, then it should be waived off, and we are also considering as well as allowing in number of cases on merit basis after thorough examination and critical evaluation of the compound product technical as well as formulation. Similarly, acute inhalation study is not required if the test substance is not included in the products which are powders or we can use by spraying. So in such cases, there is no need of acute inhalation study and that is waived off. Now CIB&RC actually has already given lot of waiver opportunities. If we see the document of CIB&RC on toxicology 2017, that is in 2017, a comprehensive document was published by CIB&RC India that is already available on the website. One can see in detail, it has provided detailed information. In this table, we can see that there are a lot of waivers and exemptions are already allowed. For example, in case of acute mammalian toxicity study, there are 6 studies, 6-pack study which are required both in technical as well as formulation. However, we can see that we have already covered, but again I am repeating that it is not required when the vapor pressure is low. That has already been discussed. Similarly, technical grade active ingredients TGI, the particle size is less than 30 micrometer. That is more than 1% on weight by weight basis. So in that case, the acute inhalation study is waived off. It is not required, and the registrant can submit their data without acute inhalation study. Similarly, the assessment of the endpoint has to be carried out and will be carried out according to the sequential testing strategies as given in the table. The assessment of dermal corrosivity, the assessment of dermal irritation using in vitro test method in both cases, as well as an in vivo dermal irritation study that is allowed for PSI, primary skin irritation study. We allow both in technical as well as formulation. In vivo testing should not be conducted. That CIB&RC accept this case where that we have already covered. If the compound or the formulation is a very strong acid that is less than 2 pH, or it is very high pH more than 11.5, then in that case it is already considered to be highly irritant and skin corrosive, and the data is not required, and the study should not be performed and conducted by the registrant, and it is not required. It is allowed, and it is allowed. The substance is then classified as acute toxic by dermal route, so there is no need of in vivo testing in such case. Also, testing need not to be required to be performed if already available information that indicates that the substance is corrosive in nature, severely irritant to the skin, that we have already discussed, and in such cases, case-to-case basis, on merit, we accept in vitro results and the data from the Agri. Similarly, if we look at the table in short-term study, that is 4 week or 3 months study, that we call short-term repeated dose tox study. For example, in 28 days study, there is no need to find out the dose in 28 days. In such cases, there is no need to conduct the study for a duration of 28 days. There is already we have covered this part, that is inhalation study, where the vapor pressure is very low, that is below 10 to power minus 2 Pascal of the particles. So ultimately, the CIB&RC has always encouraged since last many years, and we are still going further, that consult CIB&RC experts. We will help you. What are the data requirements? You need not to submit 100% data if some of the data can be exempted, and some of the in vitro or other approaches can be accepted. Waiver opportunities are there. There is no need for doing all the studies. Lot of animal killing can be avoided, and before you start the testing and data generation, it is always better, and we always promote and advise that the registrant should always consult the CIB&RC experts in toxicology, and that will help you for generation of data at the earliest without wasting resources as well as your time. That we have already discussed, there are a lot of waiver opportunities, and they are used worldwide. The most important we are discussing here is EPA. US EPA, they promote replace the testing on animals, reduce the testing on animals. So lot of policies and their suggestions are already given. If you can see, there is a guidance document for waiver or bridging of mammalian necrotoxicity. There is some guidance document on threshold approach for acute fish toxicity. Policy on skin sensitization test, then mixtures equation pilot test, What are the alternative testing framework which can be used in eye irritation potential as per EPA? How we can regulate pesticide products? Then NAM for inhalation risk assessment is commonly used now and accepted. CATMOS, COMTOX chemical dashboard, EcoTox knowledge data, knowledge base is providing lot of information for replacing testing on animals, and we can reduce it. How we can reduce testing on animals, though it cannot be completely avoided, but we can to some extent reduce, and we can save our animals. For example, acute toxicity testing and bridging guidance, acute dermal toxicity test for pesticide, which are single active ingredients. Then pesticide formulation, maybe single or maybe combination. Then neurotox battery, subchronic inhalation test, subchronic dermal test, and immunotoxicity studies. So we can combine and we can use and we can reduce the test on these parameters. Then in part 158 of EPA, Talks Data Requirement and Hazard and Science Policy Council HASPOC has already given in detail how you can reduce the testing in animals. Then there is some guidance document on subacute avian dietary test where it can be exempted and waived off. Similarly, Chemistry and Acute Toxicology Science Advisory Council CATSEC provides a lot of information on reducing the testing on animals. M010 substantially similar determination. It also helps in providing information and reducing the testing on animals. There is already, we have given the website and the link where you can get the details of all these opportunities and you can utilize. Then Canada is also giving lot of waivers and exemptions, and registrants, I think they are using it and they are considering it. So we should also consider it and go through it. For example, acute dermal toxicity waiver, then bridging of mammalian acute toxicity test for pesticides. There is a guidance document in Canada. Health Canada provides such type of opportunities, and it is already accepted in Canada. Similarly, in Australia, in number of cases, one can provide scientific argument and document based on scientific principles or the data which is already published in peer-reviewed journals in lieu of toxicological studies and detailed data. You can avoid it, rather you can provide the information which is peer-reviewed and globally accepted. So that can be, that is also, we are also accepting in India, and CIB&RC is promoting for such waiver opportunities in this country. Similarly, ECBAM or EURL, that is European Union Reference Lab for alternative to animal testing, they also allow a lot of waiver opportunities that you can avoid animal testing. You can use the documents which are given in the European Union Reference Laboratory. For example, a strategy to avoid and reduce animal use in genotox testing. How you can avoid and how you can use the animal use. You can reduce and you can even avoid. Similarly, 3 R's: replace, reduce, refine. In case of use of fish in aquatic toxicity, there is a document which is providing a lot of information and one can use it, and CIB&RC will promote it and accept it for registration. Similarly, there is a document on bioaccumulation testing from EURL, and that can be very helpful to the registrants for acceptance for waiver opportunity. So CIB&RC recently has accepted while revising the guidelines on toxicity testing as well as acceptance of tox data for registration. And we are still uh, accepting that not in exceptional or rare cases, rather we will be accepting it, and we are trying to improve it and revise it with friendly way, that we will be accepting the replacement alternatives where the involvement of animals will be reduced to very low. The animal experiments should be done only when cannot be done by any other. Otherwise, non-animal testing and the data will be accepted, which fulfills the need for registration of pesticides. But it will be accepted once you provide the full and sound justification depending on the molecule and compound. If it fits into that category, the registration can be allowed. For example, we are already accepting the replacement alternatives in number of cases. We have started accepting these approaches which are in silico. That is, these are the models that you can provide the data. There is no need of any experiment, non-animal or animal. Other experiments, that is, you can use in vitro study or ex vivo data or in chemical data. Compounds which are structurally similar, there we are accepting data bridging or read across, so we match and then we find okay, it is same and waivers are allowed, however based on the weight of evidence and scientific approach. And so we are considering all replacement alternatives which do not involve experiments on animal, and they are considered for registration by the CIB&RC experts, and they are allowed registration if the data and the information provided by the registrant is scientifically robust and it is sufficient to accept the data for registration, even non-animal approach. Recently, we have revised the tox data requirements for registration of pesticides as well as biopesticides. Now, this table gives about the 6-pack study of acute studies, acute or dermal, acute inhalation, PSI, acute irritation. Here we have mentioned the animal species, but we are thinking that why to stick to that particular animal? Why can't we allow, if the sufficient information on tox study can be given in any other species or any other alternative? If we have in vitro study, as I told you, eye study or skin irritation study or skin sensitization study, if you have sufficient information and data and the endpoint provides it's not irritant, it's not sensitizer, it's the value, it's very safe compound, so we can accept that information also. So we will be trying to improve the tox data requirements, that not only rat or mice or rabbit or guinea pig is to be used, but if the study can be done in another animal or non-animal method can be used, that can be accepted. And this will be, we are trying to help the resistance and the industry and to minimize the use of the animals. So you might see some of the changes in the near future, where we will be allowing the revised guidelines to be implemented. They have been revised, but still there is a scope to improve further, and that's why we are not implementing just now though they were in August, they were allowed that these are the guidelines which have been revised and implemented. But considering the request of various registrants in India, Pesticide Industry Association, the CIB&RC has considered this view that it should be considered and we should consider non-animal methods, other approaches, waiver request, and minimize the use of animals, taking 3R approach. Similarly, we have talked about the chemical pesticide guidelines, registration of chemical pesticide. For example, earlier we used to ask for a dog study of 3 months, or even earlier if you go 1 year study. But now we have allowed that if you submit in new molecule case, peer-reviewed international published literature, it will be accepted and allowed. Similarly, earlier we used to ask for carcinogenicity and chronic toxicity studies separately. So it's a waste of animal time and lot of money. So that we have combined, and so the animals use is minimized and the data can be generated both for chronic toxicity and carcinogenicity, and it has been combined now. Earlier, we used to ask for more than one species ecotoxicity study, but now only one species is accepted for repeated dose as well as production aviation toxicity study and acute toxicity study in freshwater. So only one species instead of 2 species, except in few cases where the compound may be highly toxic or formulation may be highly toxic. In that case, that is the only exception. Such cases we ask for. Then TI from new source, that is technical import from new source under section 9(3). That is, we do not ask a lot of data as used to be asked from earlier, but now we have minimized the tox data requirement, and we ask only for acute and repeated dose studies, mutagenicity study, only medical data, some information on human tox data from which may be from other countries, as well as a simple report on carcinogenicity and genotoxicity, what is the international status. In TIM, that is technical indigenous manufacture, under 9(3). If same technical is registered for import or formulation, which is made from the same technical is registered for import or indigenous manufacture, toxicity and mutagenicity only they are required. And even if the impurities are within limits but not toxic, then we accept the data for registration of TIM cases, only acute study as well as the mutagenicity. So we have minimized the requirement of tox data. We have reduced lot of requirements, and then this TIM under section 9(3). This is under consideration. We are looking into the then me too resistance under 9(4) category. No tox data is required for both technical in case of TI or TIM, that is technical import and technical indigenous manufacture, and formulation, that is formulation import or formulation indigenous manufacture, if the registrant establishes the chemical equivalence with the 9(3). So there is no need of any tox data and we allow registration. the grant registration. In case of LLIM, only acute tox studies are required with premix along with health monitoring studies in the user. That means we have now reduced considerably the data requirement for LIMs. Earlier, there were a lot of studies required, but that has been reduced to minimum study, that is acute tox study. In case of MUP, that is manufacturing use product, only acute tox studies are required. But we have to see here that the technical grade pesticide from which MUP is to be prepared is not registered. If it is registered, only acute tox studies are required. Then you have to provide, the registrant has to provide complete and full tox data is required for allowing MUP. So if technical is registered, then there is no need. Only very minimum tox data is required of acute toxicity. Household pesticide formulations. The registration will be given based on only acute tox studies for the pesticides in solids and liquid form, along with the data on technical. That means you have to provide the data on technical for the pesticide in solid and liquid form. In case of pesticide which are in vapor form, health monitoring study of user is compulsory and it is required. Why? Because vapor form that can affect the health of humans and animals a lot. That's why. Pesticide formulations which are used in for public health. Only acute toxicity, acute and repeated dose aviation studies, and only the acute freshwater fish studies are required. So number of studies have been avoided. We have removed, and we have considered only these studies which are essential and required for grant of registration for pesticide formulation. For aircraft disinfectants, number of formulations are used, and we are getting the registration for aircraft disinfection formulation. In such cases, only acute tox data is required, provided the technical is already registered. If not, then you have to provide the data on technical, and then formulation with acute tox data will be allowed. Now we come to the biopesticides. Again, we have allowed all the waivers, all the registration requirements, and we have further simplified requirements for registration of biopesticides. In case of microbial pesticides, for example, inhalation toxicity study is only and only required for registration of entomopathogenic or entomotoxic bacteria. Otherwise, for other no inhalation toxicity is required. That is removed. Similarly, pulmonary toxicity study is required in case of antagonistic fungi, entomopathogenic fungi. Otherwise, in other cases, no pulmonary toxicity study is required. That has been removed. IP tox study, intraperitoneal. It is only required in case of antagonistic fungi and entomopathogenic fungi. So we have further simplified. Similarly, cell culture or IV study is only required for virus. Similarly, honeybee toxicity study is only required in case of entomopathogenic, entomotoxic bacteria and antagonistic fungi. Silkworm toxicity study only in case of antagonistic fungi. Similarly, we have reduced the species requirement both avian as well as freshwater toxicity. So we have reduced one species. Earlier we used to ask data of two species. So we are simplifying further. Then, in case of microbial pesticide further, there is no toxicity data required at all for previously registered strain from the same source with same strain designation and accession number. If it is already registered strain, source is same strain designation, and the accession number is same. It is same, exactly same. There is no tox data required. And CIB&RC registered the compound microbial pesticide. Note of data is required in case of if the genome sequence of conserved region of microbial strain or microbe used as microbial pest control agent, that is MPCA, is identical with already registered strain. Note of data is required and registration is granted very quickly. If formulations are developed from similar previously registered mother culture using similar ingredient and process of manufacture, note of data is required. So we have further simplified. Now we are simplified for botanical pesticides too, as well as now we are giving all the waivers and exemptions what we are considering for chemical pesticides. 9(4) Me Too resistance. Note of data is asked for technical and formulation in case the chemical equivalence is already established in case of 9(3) resistance. So 9(4) registration is given automatically. Once you apply, you get it. In case of extract and formulation under 9(4), that is Me Too, it can be obtained from same part of the plant, same process of extraction, have identical chemical composition as 9(3) registered. In case of formulation, it should have identical CC required to be prepared from the extract which is already registered in this country. So note of data is required. Subacute studies, there is no automatic requirement of subacute tox studies in case of extract, and it is decided on the basis of acute tox study report. If the product passes the acute toxicity study report and if it is safe, then there is no requirement and we take off the subacute toxicity study and give the registration. So all the resistants, dear resistants, they are encouraged. We always ask them to consult the authority, CIB&RC expert before they go for the higher testing. Why to waste money and time and time? In case of formulation, there is no requirement of subacute and long-term studies, and they have been removed. Similarly, in case of semiochemicals and pheromones, we have simplified the guidelines. All the waivers and the data bridging and other concepts, they have been allowed in these cases also, and they have been very simplified. 9(4) cases, not of data required for in case chemical equivalence is already established in 9. In case of 9(3) resistance, no subacute toxicity study data except one teratogenicity on which we are working, and we'll try to minimize the what can be done for formulation products in semiochemicals and pheromones. Not of data required unless this formulation product is added with other pesticides. Only then case to case basis that we studied and decided. In case of Lepidoptera pheromones, these are naturally occurring compounds which are designated by 9 to 18 carbon unbranched aliphatic chain. I either ending in alcohol or aldehyde or acetate functional group, and which contains up to 3 double bonds in aliphatic backbones. This can be exempted from subacute tox study, carcinogenicity, effect on reproduction, as well as the metabolism study. But then, if their use rate do not exceed 150 gram per acre per year with GAP, that is good agricultural practices that you have to prove, and they are used in solid matrix dispenser. In such cases, we are exempting all these very exhaustive and time consuming, and a lot of money is involved in such studies, and they can be avoided and can be allowed without such studies. The registration can be granted in case of Lepidoptera pyromonas. Now, CIB&RC has considered further for registration of various pesticides, both chemical and biopesticides. CIB&RC now accepts all published literature which is peer reviewed and global literature, highly rated literature, and the data which is submitted for registration of pesticides in other countries. We are already considering it, and we will be liberal further to consider it for registration in India. Even for the compounds which are and will be registered in future for first time in our country. We have already done this favor to our registrants and the industry people. In case of change of manufacturing sites, both in India and globally, there is no need of tox data. If the registrant can establish that there is no change in the chemical equivalence of the pesticide. So, it is very good for the industry people and the registrants that if you can prove that CC chemical equivalence, you can prove it, that there is no change, it is exactly the same, whether you are manufacturing in this country or any other country, anywhere in the world, no data will be required. Now, we have already allowed lot of benefits for using the NAM, and we are still considering further for improvement and for easy registration. Now, this new approach methodologies which we have discussed, which we have rather overviewed and discussed in brief, they are fit for the purpose, they are reliable, offer better protection of human health and environment, because now we are doing lot of such practices which involves less animals for the toxicity. Go for the other studies, in vitro and other studies alternatives. These are less resource intensive and takes very less time, faster. So it helps in two ways. One, that they are economical, very less time consuming, and accordingly, ultimately, the registrant will get registration very easily and quickly. We can avoid duplication of tox data generation, because the registrant, they are submitting again and again the same data developed by different countries, generated by a lot of different laboratories, which is time consuming and highly expensive. This will help and facilitate international harmonization. So our guidelines, they are going to help the industry and maintaining the harmonization. The most important aspect of this talk is that we are ultimately going to reduce the animal sufferings. We have to use a lot of animals, and ultimately they are killed. We have to consider so many important points in this talk, and it helps if we implement and ensure using 3R principle. If we can replace the use of animal, if we can use, we can reduce number of animals for animal experimentation. And if we can ultimately refine the use of animals in experimentation, it will be the most useful and beneficial for the resistance. Replacement, reducing, and refinement. So it will cover all. Our purpose will be solved, and we will be in a win-win situation. However, not compromising the scientific justification and the acceptance of the justification and the alternative. We can consider use of all valid alternative approaches, which we have already discussed in detail, and non-testing approaches. That is consideration for waivers, data bridging, as well as in silico models, but they should be scientifically justified and in an integrating approach with fulfilling the guidelines. We are also going to accept all internationally valid non-animal methods in place of animal study data. There is no need of animal study wherever we can use all non-animal methods and provide full justification with the generated through in vitro study or ex vivo study or in vitro. If you can do it, the CIB&RC will accept it. However, don't worry, negative or positive result doesn't mean that your study will not be accepted. It will not require additional in vivo studies if you justify it properly scientifically, and it should be robust information. IATA will be applied for assessment of, for example, eye skin irritation studies and skin sensitization study, and CIB&RC will be happy to accept this approach, provided the registrant submits all the justification with the information. This we are already accepting waivers, we are already allowing exemptions, and they are allowed on the basis of weight of evidence approach. That is already known that it should be good information, it should be full of justification, that should be scientific, and the experts of CIB&RC accept it. Further, we always encourage resistance before you start generation of data or before you go ahead with the animal testing. What are the minimum data requirement and specific data as per guideline? You need not to generate data on 3 species, 4 species, 2 species. If you can do it with 2 tests, why to take do in 3 tests or 4 tests? If the data can be sufficient in rat, why to do in other species? So we have seen lot of applications where the applicant is submitting data in different species. That should be avoided, and that will be, we will be helpful to the animals. We should avoid animal I think with this information, the resistant will be able to use this uh, information what we have discussed today regarding non-animal approaches and new approach methodologies which you can use for submission of application for registration of pesticides and various types of biopesticides. I hope this will be a good informative uh, talk to the industry, not only in India, but they can use this information globally that India is not behind the international guidelines. If they are allowing, we will also allow, provided the information is submitted scientifically and it is robust with full weight of evidence. Thank you very much. Thank you.
Thank you, Dr. Jain. It's been a very extensive, elaborate, and informative, not to mention interesting presentation of yours. My name is Vipin Saini. I'm the Chief Executive Officer of Biological Agri Solutions Association of India, and I'll be moderating the question and answer session from over here. Dr. Jain, we have a couple of questions from our audience. I'll club a couple of them. Most of them have been answered and been clarified by you. Just for further clarification, will in the case where only 6-pack toxicity, acute toxicity study is required, in that case, will skin sensitization, if a person has an ITA, OECD, and other combined approach data, will that apply in those cases also?
That will be taken into consideration. Now, we have already included that alternatives will be accepted, so we will consider case to case basis with full justification if provided by the
Right, sir. And so you mentioned a little bit of QSAR data. Will other models of QSR model like Derrick, Sarah, and Vega also be acceptable during submission of data?
In some of the applications we have already accepted in silico models, particularly in cases of export cases. We have already accepted the information provided by the applicant in silico models, where the compound found is it is not causing cancer, or it doesn't have potential for neurotoxicity, or it doesn't causing any reproductive problem, etc. There we have considered in silico model. However, we will be taking into consideration the use of in silico models, provided the information is scientifically robust. Otherwise, they say carcinogenicity study or neurotoxicity study or immunotoxicity study simply in silico models sometimes do not provide sufficient information which can be accepted without animal study. So it depends case to case basis.
Yeah, thank you, Doc. Thanks for the clarification over there. One other question has come across regarding that. Is there a possibility of waiving acute dermal toxicity study in rats in case a person has dermal and eye irritation data on rabbits? Is such an opportunity, data waiver opportunity available?
I hope so, that we will be considering in future. Till now, we don't have such opportunity, but if the resistant applies and asks for considering instead of rat, rabbit may be considered. So it will depend what are the results, and if it is safe, I think it can be considered.
Thank you, Doc. Thank you. Doc Saab, there is one question you raised about chemical equivalents. In such cases, then total toxicological waiver is probably possible. I would just like to ascertain whether there is a specific kind of analysis wherein the chemical equivalence is done. Is it a single batch, 2 batch, or 5 batch analysis which will be acceptable and qualify a person for total data waiver in this regard, as respect of say safety evaluation is there?
This aspect is already under consideration, and a committee is there which is considering this regarding 5 batch analysis, because in India it is not so common in this country. In other countries it's very common 5 batch analysis, and they are giving registration without data. We are also under consideration that if you submit like this, so this is going to be applicable in general. But
That's,
5 batch, what I think.
Right. Doc, I have I mean, you did mention something about, recommended waivers from OECD and other countries like US and Canada. You've given a brief slide on that. But I would just like, I was just very curious to know that whether additional waiver opportunities based on retrospective analysis conducted by them will also qualify as data waivers over here in India while registering pesticides?
Yes, it is under consideration, and I think it will be considered if you give the information already available, and if they have allowed waiver, whether it is EPA information or in Europe or in any other countries. So if that fits in our case, we can consider and allow waiver. And in some cases we are allowing.
Yeah, thank you, Dr. Doctor, one last question I'd like to pose before we wind up the session. It is, oh my God, we've got a number of questions coming across. Just one question I'll be taking up after this is, what the CIB and RC also consider internationally valid refinement and replacement approaches for ecotoxicity testing, such as the case may be for fish embryo toxicity test, which qualifies as OECD TG236, and another OECD TG249, which is for acute RT-GIL W1 cell line. Will such kind of tests also be, considered across?
I hope so. In future will be considered, but we will take it to RC, and RC will make, I think, policy to accept all OECD valid TG studies that you have mentioned, and they are under consideration, and perhaps they will be accepted.
Okay. Sir, there's a couple of questions now pouring in. This just shows the kind of interest the audience has in your presentation and the kind of, labor you've placed in the presentation and made it so interesting. One of the questions has come across that in case of acute oral toxicity in birds, it is only one species, irrespective of whatever we're talking about, because this will qualify as ecotoxicity, right?
Yes.
Right. And so there is this query which has come across. I believe the person should take it across with you separately. Is livestock study still required for registration?
It is still part of a new molecule.
Only in case of new molecules?
Yes. If the molecule is already registered, I don't think any livestock study is required. You can submit the literature already available of that compound in livestock, whether it is in buffalo, cow, or goat. I think it will be accepted. Right. And I have accepted number of cases.
Right. Sir, another thing that these new guidelines which have come across, will they also be applied across the applications for registration already submitted to the government, or no, they will be, I mean, perspective in that regard?
No. they will be implemented prospectively once all the guidelines have been thoroughly revised, considering all the queries of industry and other applicants, and that's why we have deferred for implementation. Mm, but I think they will be soon implemented in near
Right.
future.
Dr. Saab, I'm sure I'm slightly confident that carcinogenicity in mice is not required any longer in India, but could you just clarify for the sake of the audience over here, whether it is required or not in any case?
No, it's required only in new molecule.
It is carcinogenicity mice is required only in, sir?
New molecule for registration. New molecule.
Okay, so rat and mice is both required for new molecules, right?
Yeah, for new molecule.
Right.
Once you get registered new molecule, then no need in future.
Right. So this question has come across one very interesting one, I'll say. Is there any future plan for a policy to be implemented before doing any vertebrate testing like in other countries with permission of the regulatory authorities over here in India?
Your suggestion is very good and it is welcome, and we will frame a policy.
That's very kind of you, sir. Very kind of you. Sir, I believe for the benefit of the audience, this was a common query that came across whether the next slide of Dr. Jain will be available. This recording will be available online. So therefore, you can always go online to the PETA website, or we will also try to share it to the maximum group and all the audience
Yes,
that
yes.
we can reach out
Will
to.
be available. You can listen, you can read, no problem.
Yeah, that is the beauty of having a recording, sir. So thank you very much, Dr. Jain, for your wonderful presentation and all the queries that you've been able to clarify with such ease. Ladies and gentlemen, if there are any other further queries that you may have, please do write across to Dr. Ankita Pandey from PETA. I'm sure she will direct all your questions, and necessary answers will be communicated to you in time.
Most welcome.
Uh,
You can have any query anytime, no problem. I will answer.
Yes, sir. Yeah, that's right. And ladies and gentlemen, before we wind up, just a piece of information. Post this webinar, you may receive a pop-up for a survey to be conducted on NAMs on this webinar. Please do spare a couple of minutes of your precious time and do give us a feedback on how this webinar has gone across. How do you feel? What are the kind of improvements that we may come across? And together, all of us trying to bring these webinar series over to you will make it a point to come across to your expectations. Ladies and gentlemen, thank you very much for everything. Have a wonderful day. You've been a wonderful audience. Thank you very much. Stay blessed. God bless.
Thank you.